Okay.
This is where we left off here with good, bad, and ugly. Is that right?
We just finished that.
Just finished it. Okay.
Oh, you should have closed the door and just gave us a free day.
I should have locked the door. These are combination terms down here at the bottom: glycoproteins, proteins, carbohydrates mixed, lipoproteins, lipids and proteins combined, ketone bodies caused by rapid breakdown of fat, which may occur in uncontrolled diabetes or a strict low-carb diet. And of course, if it's out of control and continues, could lead to ketoacidosis, ultimate death. Now, give me some symptoms of diabetes.
Symptoms,
Yeah, just
Okay.
symptoms.
Low blood sugar?
that's on the low end. High,
That's
you have
after you do a blood test. What about just everyday symptoms?
rapid heartbeat at times, no
And
energy
afraid
at
to go into
times.
coma,
Do what? Yeah, oh yeah, that's what it's talking about right there. You can go to coma.
cause they didn't know he was diabetic.
Yeah.
Yeah, so before he knew, he went to a coma.
Well, don't you have constant thirst?
Yes, constant urination also.
Constant urination with it also.
No energy.
Lack of energy.
Your eyesight,
You can lose eyesight pretty quick.
Poor
It's-
circulation.
You'll have poor circulation. Wounds don't heal as well.
You'll, might lose extremities too.
Tingling, that's part of the poor circulation.
Do you have
What's
some swelling also?
Oh yeah, you can. Guess I'll take roll again later. Just kidding. What's a symptom? Let's say you're talking to this person and you're just right there. What's a symptom you may pick up just talking to them?
The breath.
Sweet smelling breath?
mm,
I'm not sure if it's a pleasant, sweet smelling breath, but they say a fruity breath. I'll have a test question, something like that on there, but, it's not juicy fruit, we'll put it that way. It'll knock you down. If anybody's ever done the probably more the Atkins diet,
Yes.
you reach that point of that nasty breath, and it's called ketosis. And that's the point in that diet at which you level off and start sustaining where you're at. But what you've done is created a place where the body starts burning fat pretty rapidly, and that's where your weight loss comes in that program. It's using energy to break down food that's hard to break down. That's the whole idea of the program. So when you reach that point, when you wake up and can't stand your own breath, you know you've hit ketosis. And that's where you level off and try to stay there and maintain your weight. And that's a controlled state. I'm not sure it's a totally healthy state. I mean, Mr. Atkins died pretty young. The South Beach diet is a slower, twisted version of that, where you have more vegetables and stuff in the deal. It's gotta be a little bit healthier. It's gonna be a slower process than a weight loss, but a longer process. And all those try to do is change your habits in life, where you don't eat everything that you pick up and look at everything and wanna eat everything you pick up. And, that was, those combination terms, and kinda give us an idea what to look for there. All right, the last category of organic molecules is proteins. And proteins consist of carbon, hydrogen, oxygen, and nitrogen. Notice the first two just had carbon, hydrogen, and oxygen. Proteins have an element nitrogen that no other organic molecule has. I usually point that out. It's not necessarily a good thing, and typically they're waste, like urea, uric acid, creatinine are all waste products of breaking down proteins and dealing with nitrogen. The 20 necessary amino acids, keep that number in mind, because we're going to see it before we get finished again today. And of those 20, we make 11 or 12 of them, we have to consume 8 or 9 of them. And consuming 8 or 9 is not a difficult trick. If you like meat at all, a small amount of meat, a couple times a week gets you plenty. Most of us overkill and say you need 6 ounces of meat, and we'll just do 12 while we're at it. Get the big steak while we're at it, right? And I don't know that that's necessarily healthy, but we certainly get enough amino acids out of it.
It works.
Yeah, it works. You just really shouldn't do that 4 or 5 times a week. Now, proteins are used for enzymes, part of metabolism, catalysts, part of metabolism, antibodies, your immune system, carriers, getting things throughout the body that needs to go places. And that could be hormones. If hormones are steroid based, they're not gonna mix with your blood very well, so they have to get on a protein and be carried where they're going. And then buffers, maintaining pH. So I think the point that we need proteins in our diet is very well made here. Now, any vegetarians in here? Yeah, I don't find many vegetarians in southeast Oklahoma. I mean, we're kind of shoot something and eat it kind of deal around here, you know. Doesn't matter if it's in season or not. What season? We can't read rules. Laws, rules, what's that? We can't even read. So occasionally I'll run into a vegetarian, and I mean, full-fledged vegetarians that have studied what they're doing, they can fulfill all the essential amino acids. Most people say, oh, I'm just gonna quit eating meat. And if they don't know what they're doing, one of these capacities is gonna start failing. Usually it's immunity. And you say, well, I mean, they're not admitted into the hospital, they're so sick. It could reach that point. But if they're gonna decide they're gonna use refried beans to fulfill all their other amino acids, they're gonna miss a few. They're gonna need some out of wheat, and some out of rice, and some out of beans, some out of peanut butter. Nice combination of that. I guess you could mix all that together. Every meal, little side dish of this would work just fine. Does that sound good for breakfast? Beans, rice, what'd I say? Wheat, throw some wheat flour in there,
Peanut butter.
and peanut butter. Well, that ought to stick to your ribs, hadn't it?
I know a friend that lives in California. He's a bodybuilder, and every morning, peanut butter, beans, rice, and tuna. Just
Oh, well, see, he's not a vegetarian by any means if he's slapping tuna
Yeah,
down
but
the hatch. But
talking about
if you
nasty in a
mix
bowl.
that in there with tuna, that would add a really unique twist to that, wouldn't it?
it's not great.
But what happens is they'll get stuck on one of those. They'll get stuck on peanut butter, or they'll get stuck on beans, and they're missing 2 or 3 essential amino acids. And you say, oh, well, we just need 20. I got 17 of them. It's not good enough. We need 20. We don't need 17 of 20. We need 20. And a lot of times their antibodies start falling a little bit, and they'll have a few more colds, or they'll catch a bug that nobody else does. You know, it seems slight, but it begins to get major. So they have to be extremely cautious and knowledgeable about what they're doing to maintain what they should. Us meat eaters, it didn't take much. We got it covered. Our problem is it's clogged vessels, and our immune system don't get to where it needs to go kill something. All right, moving from there to nucleic acids. And this is, in fact, I'm gonna put up the other, went straight to that, because getting right into the nucleic acids here. DNA and RNA, and what's going on with them. So this little section is how DNA makes DNA, and DNA makes RNA, and DNA makes proteins. And if we compare DNA and RNA, the good news for us is that DNA is in control. When DNA is in control, it pretty much does its job correctly. If it makes a mistake, it has a check system. It has a backup check system where it'll go back and say, oh, messed that up. It'll cut it out and redo it. So we don't have tons of mutations, although each generation has at least one new mutation, which makes y'all more of a multiple mutant than me. But it could be good, who knows. In animals, typically mutations are bad. We don't tolerate mutations very well. We're plants, we mutate plants right and left. It's crazy what we do with plants. So we try to minimize mutations in all animals, and DNA does that for us. Things that are RNA controlled, which are a handful of viruses out there, those are not good viruses at all. And you're probably thinking there is no such thing as a good virus, but yeah, actually there is. There are viruses that are actually harmless to us that we've used in medicine to actually be gene carriers into a cell. And so we can do gene splicing and things like that using viral carriers. Those are good viruses that we can use, but viruses that are RNA controlled are called retroviruses. Retroviruses are viruses that tend to change. And RNA in control, if it has a check system, it doesn't use it. It makes more of itself and screws up all the time, and it just has a poor attitude about its job. And so what we end up with is the same organism causing the same problems, but slightly different all the time. A good example of this is the AIDS virus. Have we ever got a grip on the AIDS virus? It's still out there killing people like it used to. We've figured out ways to slow it down, medications to keep your immune system up, but we found nothing to slap the AIDS virus in the face with. So we ought to create a vaccine. You can't create a vaccine for retroviruses. They change too much. By the time we get one to match it and get the FDA to prove it 5 years later, the AIDS virus was changing itself 13 times in 1 year. That was at last count.
Isn't the flu virus also retrovirus?
Well, there can be some retroviral flus, but there's so many different variations of flus. The flus that come through this year could be just a slight change from last year, but it's slight enough that it can make you sick again. See, at last count, there's over 150 different kinds of flus and over 150 different kinds of colds, which is a cousin to the flu. It's just slighter. So, to say, here's a flu vaccine and it's going to keep you from getting the flu, our flu vaccine is a crapshoot every year. Our flus come from Asia. They come by way of bird of wing. And when we're in the summertime here, Asia's going through flu season over there. So what we do is look and see and try to isolate the major flus that's attacking them. And we take the top 3 flus that's attacking them, then build a vaccine and have it here by October to give us immunizations against it when it gets here. Sometimes we get it right, and sometimes we miss all 3. And we darn near missed all 3 this year. I think there's been several people diagnosed with the flu this year that had a flu shot, but what they're seeing, it's minimized. It's not as bad. So your body had enough immunity against something similar that it could still attack it and lessen the effects. But the flu's killing people this year. So Tamiflu's been a nice drug to come out. You get it in the system in the first 48 hours and it turns the course on it. It slows replication of it and allows your body to catch up faster. So it's done a wonderful job this year. So, but it's not supposed to be prescribed to small children though. I think under 4, under 6 or something like that.
6, I think.
I'm not sure why that is, but I've seen it prescribed to even younger children than that. It's like, do you give it to them? I mean, does this doctor really know what he's doing? Doctors kill people all the time. So anyway, we're going to spend quite a bit of time here today with DNA and RNA. Here's your comparison of the two. DNA is a double helix. RNA is a single strand. DNA is going to make itself, and then it's going to make RNA. DNA uses a sugar called deoxyribose. RNA uses ribose sugar. And DNA uses a base called thymine, and RNA uses the base called uracil in its place. The other 3 bases are exactly the same. So the nucleotides are the building blocks of the nucleic acids, and they consist of sugars, bases, and phosphates. Not bases, but bases. All right. So we're going to look at these 3 terms. Replication, transcription, translation. Replication's DNA making DNA. Transcription's DNA making RNA. And translation is DNA making proteins. And that's going to be by way or via a messenger RNA molecule. With those in mind, let's look at a little bit of history of this. Who discovered the structure of DNA molecule? Anybody know? They were Nobel Prize winners in 1965. Must have missed that in the reading, huh? You should remember this from zoology. Did you pass that class? Something's wrong with that picture. Zoology. Who discovered the structure of DNA? She's saying, who cares? That's what I'm seeing on her face, is who cares? Well, if they were still alive, they would care. Watson and Crick. We're just not getting any brain activity this morning. But it's familiar that you hear it, isn't it? Watson and Crick discovered the structure of DNA. And I'm not sure how much they actually did, but they won the Nobel Prize. From what I can understand, by the time they got out of bed in the morning and walked their dog and drank some coffee, they was ready to eat lunch. And then they worked on the model, the DNA model, a couple hours a day. And won a Nobel Prize. You know they had to be on some kind of government funding grant of some kind to do this. But there's other guys working on this, and they, a couple other guys named Pauling and Corey, actually wrote the first article on the structure of DNA, except it was wrong. And Watson and Crick knew just enough to prove it wrong. Well, they kicked it in high gear then to go ahead and finish off their job, and they worked at least 3 hours a day after that. And within a couple of weeks, they wrote a rebuttal article that disproved Pauling and Corey what they were doing, and went on to say that DNA molecule is a double helix molecule. Now, they didn't do that by themselves. They had other people help them. A guy named Maurice Wilkins described the backbone of DNA, which is phosphates and sugars. A guy named Irwin Chargaff described the bases and how the bases always match. And Chargaff's rule says, and he listed the bases in 2 categories, pyrimidines and purines. Chargaff's rule says the pyrimidine cytosine always matches the purine guanine, and the pyrimidine thymine always matches the purine adenine. In other words, adenine and thymine always matches, and cytosine and guanine always matches. That's Chargaff's rule. Now, Watson and Crick didn't come up with that, or Chargaff did. And then probably the one that really made this whole deal, that gave them the Nobel Prize, was a lady named Rosalind Franklin. And her specialty was x-rays. And she was actually able to take an x-ray of the DNA molecule at such an angle that it cast shadows on the x-ray. And they could determine that it was not a triple helix structure, like Pauling and Corey said, but it was a double helix structure. Now, granted, that was pretty serious information, and Rosalind Franklin was up for a Nobel Prize just a couple of years after they got it. But she died at 36. Radiation exposure, cancer killed her. Now, look what we've done with her knowledge today. We've got more scans, we do more stuff, we can see more internal parts of your body today than ever before. There's no telling how much x-ray she is bombarding herself with every time. You know, you find a new toy, you want to play with it all the time, she's just killing herself right and left unknowingly. But because of that, look what we have today. So, leading into these bases, if I were to give you a DNA sequence of bases, A, C, G, T, T, C, A, G, A, T, you would need to be able to match that. And it's as easy as this one. A and T's matches and C and G's match. I mean, it's easy as that. It's multiple choice. You match the A and the T and the C and the G, and continue right down, and you got it. So there's only 4 bases. If you can remember A, T, and T, the other 2 go together as well. So it can't be that hard. That's based off Chargaff's rule. Thus, A and T always match, and C and G always match. If we produced a strand of RNA from the original strand of DNA, you'd replace one base. What base are you replacing? Thymine. You simply, anywhere you would normally put a thymine, you put a uracil, and you just made an RNA strand. It's as simple as that. So here with this sequence, here's an A, you'd normally put a T there in DNA, wouldn't you? With RNA, you put a U. That's it. Go down, find another A. There's another A, you put a U. There's another A, you put a U. You got it. So you've made an RNA strand according to Chargaff's rule. And of course, the last thing simply says that uracil replaces thymine in RNA. Now, with that said, we're going to replicate, transcribe, and translate. Replicate is DNA making more DNA. So as they describe the DNA molecule, it's a double helix molecule. And we're going to have some bases matching up here. You have sugars, phosphates, and a base. What is that? What consists of a sugar, phosphate, and a base? A nucleotide. Yep. Something we just talked about just a few minutes ago. So all this is actually happening in the nucleus of the cell, and some of it's going to go out and happen in the cytoplasm of the cell. Now, when we look at the cell cycle, probably Monday, in the cell cycle, we looked at metaphase and prophase, metaphase, anaphase, telophase in the lab. The cell cycle consists of one other phase, interphase, at the beginning of it. All this is happening in interphase. Interphase is one of those phases that served not that long ago, really, but a few years ago was thought to be a resting stage of the cell. And with increased technology and more study of the cell, it was far from that. There's more going on in interphase than any other stage of the cell. Interphase's purpose with what we're doing, replication, transcription, and translation, is to double the information of the cell. So when we get to mitosis, when prophase, we start organizing the genetic material. We organize it and then we separate it equally and then divide the cell. Now we have two identical cells from the original mother cell. If you did not double the genetic information, you couldn't do that. You can't just split it in half and keep splitting it in half. And I mean, if we have 46 chromosomes and your kids have 23, what's next? I mean, before long, humans are gone, right? So in meiosis, we have to do this. We have to continue to produce the right amount of chromosomes to continue the same chromosome number in that species. So that's where we're at here. We're going to make a DNA strand, and I'm just going to do 3 because 3 is a significant number, what we're working with. But they'll tell you that a sequence of 10 base pairs, there's a twist in the DNA molecule. And it's a spiral staircase is how they describe it. But if you have a sugar and a phosphate, which Maurice Wilkins described as the backbone on both sides, sugars, phosphates, and a base, you have the nucleotides. Nucleotides are building blocks of nucleic acid. So if all these are connected, we have a nucleic acid. Now, with that said, in the nucleus, the DNA molecule will unzip itself. There's usually a command, some type of a enzyme that commands this process. And in this case, it's probably DNA polymerase, I believe is the one it is. And it starts unzipping. Well, what's unzipping is these little hydrogen bonds right here. It's strong enough to hold it together, but weak enough we can take them apart to build more of the same. That's all we're doing is building more of the same. So here we have an adenine and a thymine, but they're not connected. The bond is gone. We have a cytosine and a guanine not connected, and a thymine and an adenine not connected. But in the nucleus, there are free nucleotides prepared to join up. So as free nucleotides come cruising through, we see a thymine add to that adenine. We see a guanine add to that cytosine, and we see an adenine join to that one, and on the opposite side, we see similar. And then we end up with two brand new strands of DNA that looks just like the original strand of DNA. And we've doubled the DNA genetic material. Now we're prepared to create chromosomes, organize the chromosomes, separate the chromosomes equally, and divide the cell equally in mitosis. Okay? So that is all there is to replication. Now, in the process of transcribing or transcription, in transcription, when we unzip the DNA molecule, DNA is still in control. The RNA is a single strand, not a double helix. So when we transcribe a messenger RNA, we'll use one side of the DNA molecule or the other. In this case, what will join with adenine and make an RNA? Uracil. Very good. Still have guanine here, still have adenine down here. Once that's made, that RNA molecule, which is messenger RNA, once it's transcribed, it unzips off the DNA and heads out to the cytoplasm. It goes out to the cytoplasm and joins to a ribosome. Anybody know what a base definition of ribosome is? From zoology, Kelsey?
It's
Well,
been-
retroactive, you're great. What happens on the ribosome in a cell? I know y'all had zoology, correct? Whether it was me,
It's
Miss
been a
Brooke,
while.
Mr. Brown, Miss Afzali, somebody had zoology in here. And we still don't know.
It's been a while.
Been a while.
What was it the first of this semester?
Last summer.
Oh,
Yeah,
it was a-
that's
do
your
what?
point.
Protein's made?
Yeah, that's just sign of protein synthesis. Everybody else is thinking you cheated, but you actually got a little brain activity going there. Good job. So if we transcribe the messenger RNA and it carries its code of UGA, I'm going to use dots for a bunch more of those, that attaches to a ribosome, we're preparing to do what? Make proteins, right? We're preparing to translate the information from the DNA into the proper proteins. Now, I use these three for a reason. How many amino acids must we have? 20. We remembered that number from a few minutes ago. That's good. That's why I started trying to mix this in the same lecture. In zoology, it's a few weeks apart, we forget the 20. 20 amino acids. Now, if each nitrogen base, there's only 4, represent an amino acid, we're a little shy of 20, aren't we? So the scientists got to calculate, well, maybe there's combinations of 2 of these that represent a nitrogen base. And if you use combinations of 2, you can get a whopping 16. It's like we said a while ago, 16 is not 20, or 17 is not 20. We got to have 20. So they said, well, the only other option is combinations of 3, which is a little bit overkill because we get as many as 64 different combinations. But the overkill is this. It is margin for error. It gives our system capabilities of making mistakes, yet they're not mistakes. Okay? So this is called, and this being the messenger RNA, the messenger RNA carries what's called the codon. The codon. Also known as a triplet code in some books, but codon is what we will represent that with. And the transfer RNA carries the anti-codon. Think that's going to be a match? Probably. I'm going to draw this little funky transfer RNA here. What matches U in RNA? Very good. What matches G in RNA? All right. What matches A in RNA? Excellent. Some of you got it right, some of you didn't. Put an L in there, would really stir up all kinds of steam, don't we? ACU matches UGA. What that match does is create an amino acid. amino acid comes down here and hangs off the ribosome. That transfer RNA, and some books actually refer to it as a taxi, gets out of the way. Another transfer RNA comes down and sweeps this messenger RNA until it finds its 3 that matches it. That creates another one. Another one comes through. And it continues until it reaches the end. Now there is a triplet code that represents end chain or stop. How long is a normal polypeptide chain, which is a protein? The minimum is 5. The average is about 500. Minimum's about 5, the average is 500. So let's say we stopped right here. Can anybody name some amino acids? You know any amino acid names? Y'all don't know a whole lot today, do you?
Friday.
It's early Friday.
You do the same thing to me Monday, Wednesday.
Well, we can say it's Friday on Friday.
Mondays on Mondays, Fridays on Friday.
Monday, we're still in neutral. Friday, we've already gone into neutral, but we got nothing done Wednesday. So what's our excuse that day?
I can find one.
I know, it's 9 o'clock in the morning,
That's it.
way too early.
Exactly.
We're trying to get over that hump for hump day. Trying to get over it.
Every day's a hump day, for you, Dave.
Yeah, that's true.
And I, honestly, I feel a little bit sorry for you,
Why?
because you have to deal with the coach you have to deal with here. Is that understandable?
That's very understandable.
Okay, so to finish the rest of my points here. There's 20 different amino acids. Let's say, glutamic acid is represented by AUG, OG. Yeah, we're just making something up here. Now, the Zoe book had one of these charts in there. It's a pretty cool chart. I need to make copies of it for here. Now, if you look at one of these charts, it may have a CG, it may have a AG, and all those give you glutamic acid. All of them could give you, that's part of the margin for error. Each one of these little amino acids may have 2 to 5 different options to get the same amino acid. All right, what if the glutamic acid, or what if the amino acid valine comes in here at a GG? That's close, but it's a different amino acid. And we're trying to build a protein chain that we all have called hemoglobin. And in the protein chain hemoglobin, if you replace glutamic acid with valine, you no longer have hemoglobin. You have sickle cell anemia. So you can see we could make some mistakes and still get glutamic acid. One other slight mistake and we have sickle cell anemia. So the margin for error is huge, although still we have the chance for a mutation. And that's a serious mutation. The only advantage, well, it's up here now.
I'll get it.
The Whopper made it to the front.
I'll get it.
The only advantage of that mutation would be the fact that you, with sickle cell anemia, would never get malaria. Malaria has to have normal hemoglobin to live in. I said, well, that's good news. Not really, you're still gonna die early. So if you have normal blood and live in a malaria area, then you can get malaria and die early. But if you're gonna live in a malaria area with sickle cell anemia, it won't be malaria that kills you. It's much better to have normal blood and move out of the malaria area. Get out of there and survive that. So you can see how easy it is for a mutation to occur. Yet, look at the huge margin for error that we could have made a mistake, yet no mistake was actually diagnosed or seen. Gives you more to worry about now. See, the more you learn, the more you have to worry about. Which anybody have any questions over this? Do you know where to start to ask questions? You still lost? A little bit of light peeping through?
Too much muddy water.
Muddy water. That's better than no water at all. You always gotta look at the positive side of this.
How
So-
much of this are we gonna need to know?
All of it, okay.
Cool. Oh, I won't make you do any drawings. I had to do drawings in
What would a
college.
question be
They
over
were
this?
brutal, but do I?
What a question be over there.
What kind of question?
Yeah.
I'm gonna have you matching A's and T's and C's and G's. Not only on test one, but on test two, just make sure you remember it.
If I remember
I can do
correctly,
that.
this is DNA, this is RNA. Which matches to make correct? That was zoology.
I will give you, well, if you can do that, you can do this, so.
Mhm.
If I give you a DNA sequence down in the multiple choice, you're gonna have to pick the one that matches.
Oh, that's not so bad. We can do that.
That's pretty easy. You'll need to know definitions of these. This one's DNA making DNA, transcription, DNA making RNA. And we're starting to breathe. Look, people's getting notes out and starting writing stuff down. I said test, holy smokes. Translation is making RNA from DNA. Now,
The very last you don't know where was like translation.
this is translation. We translated these amino acids from the messenger RNA, which came from DNA. We translated that information from DNA. And we want DNA to be ultimate control down there because originally right here, what if a cytosine come in right there and attach? That's not even supposed to be there, is it? DNA's check system sees that, uses a cleavage enzyme and cuts it out, tells it to do it again. So it can correct itself.
Spell check.
That's what it does. Now, will it miss some? Sure. But it fixes a lot. So that's why we want DNA in control and we don't want retro in control. We don't want RNA running this deal. Now, questions you might see other than straight definitions. It would, in physiology, it would be too simple for me to say DNA making DNA is.
Replication.
That's good.
Very good.
That's real good. That question will not be on my test.
But the word physiology itself is hard enough.
Yes, yes it is.
Even
Have
say.
you practiced spelling that?
But no.
Here's the type of question you'll see. You'll see off of these words. And I had problems with questions like this for a while, but I went back to them because I think they have something in them that makes you think. Let's say Fred was diagnosed with extreme muscle buildup, beyond normal. And the doctor figured that for some reason excess protein synthesis was occurring. Well, which one of these am I talking about already? Translation. So the doctor prescribed, and I make up drug names that don't exist in the market. Doctor prescribed blah, 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 that tends to slow down the progression of excess protein production. What is it stopping? What is it slowing down? Transcription. So when you see protein production, you should already connect it to that term. If we're slowing down DNA replication, then we're connecting it to replication. If we're slowing down the process of RNA carrying information because it's happening too fast, then that would be transcription. So, you know, I make scenario questions out of these to make you think through the process. Dumbed down definitions don't always make you think to make it stick. It's like, I should have asked some crazy question about Watson and Crick. Then you'd have remembered it. But I probably said, who discovered the structure of DNA? Watson and Crick, I got it right. Now, turned that off and never remember it again. So when I ask you the next semester, I don't know. It's got to stick to you a little longer than that. Slap peanut butter on it, it'll at least stick to you for a little bit. Alright, so that's your DNA and RNA and protein production right there. The next page, if you flip to your notes, starts with chapter 3. And chapter 3 is just a cell, and then it's gonna lead into mitosis, cell cycle, and all that kind of stuff. So we break the cell down in 3 components. We have the cell membrane, we have the cytoplasm, and the nucleus. The nucleus is easy. I mean, that's the control center. We just said that's where DNA is at. If I ask you a test question and say, true or false, DNA is always found in the nucleus. It's true. It's always found in the nucleus. It's never found outside the nucleus. The nucleus is where it's at. True or false? RNA is only found in the nucleus. It's false. Messenger RNA is made in the nucleus, but doesn't stay there. It heads out to the cytoplasm where it joins up with ribosomal RNA that connects the ribosome and the messenger RNA, and the transfer RNA that's carrying an anticodon. And they come up with precursor messenger RNA and precursor transfer, I don't even know, they come up with all kinds of stuff now. So, if we start here with the plasma membrane or the cell membrane, then we're looking at a phospholipid bilayer that is described, and they've got about 3 different ways they describe it. They all mean the same. It's semi-permeable, selectively permeable, and differentially permeable. All means the same. The cell membrane is picky in what it allows in and out of the cell. But if you need something, it knows how to get it in there. If it needs it out, it knows how to get it out. Now, chapter 6, we'll get into more of how things go in and out of the cell, which is part of test 2. What we have here is the phospholipid bilayer. Now, the 2 layers, embedded proteins throughout it. Those proteins are used for carriers. That's one way things get in and out. Hops on the protein, carries across cell membrane. But what we have here is, these are the lipids. Lipids are made of heads and tails. The heads pointing toward the intracellular fluid and the extracellular fluid, and the tails are all in the interior. The heads, our lipids are different than the average oil in a bottle. You mix oil in a bottle with water, what happens?
Eventually
They
separate.
don't like each other, absolutely hates each other. Our lipids, the way they're turned, it's almost like they're inside out. The heads are hydrophilic, they love water, and the tails are hydrophobic, they hate water. So with the heads being hydrophilic, water comes right up next to the cell membrane, whether it's in the outside or the inside. They love it. So our lipids are not the average lipid. The tails hate water, so that deters anything passing through, because it's a repellent. And that's what makes our cell membrane unique. Now, the book may use reference to the fluid mosaic model as the modern cell membrane theory. And you may run into that term as you read through the chapter, which I'm sure most of you already have, right, Kelsey? And the fluid mosaic model, mosaic means random. It is basically this in a word description, is all it is. Steroids would readily pass through a cell membrane. Why? Lipids and steroids are virtually the same. They mix. So, like drugs that are steroid-based, albuterol, inhalers, things like that, if you have an asthma attack and you were taking a medication that take 30 minutes to work, wouldn't do you a lot of good, would it? So these are all steroid-based because we know it gets in the system and does what it's supposed to do within seconds. It can readily pass through. Don't need help. It gets there. So this bottom part's telling you what I just already said. Hydrophilic, hydrophobic. Hydrophilic's water-loving, hydrophobic's water-hating. And that's how it's made up. All right. Mark that endocytosis there. We'll start up there on Monday. And we'll ease on through this. Let's see. Monday, Tuesday, yeah, that should be next Friday. We can make that easy. We'll have Monday, y'all in Monday's lab or